Thanks everybody for coming. So today I'm going to be talking about publication titled Bacterial Quorum Sensing and Metabolic Incentives to Cooperate, which was published by Ajay Dandekar, who is a professor at the UW, and you can actually contact him online. He has his email, so if you guys have any questions or want to reach out to him, that information is available to you on the UW website. He's also a practicing physician, so outside of microbiology, maybe you have questions on his career or how he got there. I think he'd be more than willing to maybe answer your questions. It was also written by Sudha Chugani and E. Peter Greenberg. So I'm just going to start with a general abstract, and I'll get a little bit more into some context and some general ideas that we could probably expand on a little bit, but I just wanted to introduce the publication to you guys a little bit first. So the main focus of this paper is based around this bacteria called Pseudomonas aeruginosa, which uses quorum sensing to control production of goods being proteins and enzymes that are collectively used by bacterial community members. So a lot of the time bacteria grow together, and through quorum sensing, Pseudomonas aeruginosa is able to share products with each other to help grow. And it's quorum sensing is very beneficial in a certain population. Social cheaters or mutants are able to take advantage of cooperators. Social cheaters being, so in quorum sensing, there are bacteria called cooperators and bacteria called social cheaters. So there's two classifications of how they act in a community. Social cheaters are those that'll uptake all of the resources that are shared by this community of bacteria, while cooperators are the ones who are producing these goods. They use it as well, but they also put back into this community of bacteria. So social cheaters, they don't produce any of those enzymes or those proteins that can be shared by all the bacteria. They only uptake and are therefore sort of cheating socially. They refer to them as mutants in the paper because they don't, they're technically mutated not to produce certain enzymes and proteins. So these enzymes that I'm talking about, these goods that are shared by the community, are controlled by quorum sensing signals that can help constrain social cheating, and being able to regulate social cheating provides for a more cooperative system. So a little bit more on what quorum sensing exactly is. It sounds really weird and fancy, but it's really just a type of cell-to-cell communication that allows for bacteria to monitor population densities and consequently activate specific genes. So if you look in this picture to the right, there are these little red pentagon-looking things. Those are chemical signals. This is the form of communication that the bacteria use. So these little chemicals leave the cell and re-enter cells and reach a receptor. And if there's enough of these chemical signals, the receptor will send messages within the cell to activate specific genes to produce like an enzyme or a protein that will eventually leave the cell. So the population density part of this is that if there's a large enough population, there will be more signals, and once the signals reach a certain threshold, that's when the receptor actually starts activating specific genes. So they don't immediately activate those genes once they receive a signal. There has to be a certain amount reaching it first for it to actually start really activating those genes. So this system of quorum sensing of signal sending and receiving controls the production of goods, which again are enzymes and proteins that are shared by a community of bacteria. And this is controlling both public and private goods. Public being enzymes and proteins that'll leave the cell that can be shared by all of the bacteria in a community, and private being those enzymes and proteins that you use, that a bacteria might use individually, and they don't send it out of the cell for other bacteria to use. So just a little bit more about those little signaling, those little red protein chemicals, or sorry, pentagon chemicals that we saw in the last picture. In this particular research, they focused mostly on acylhomoserine lactone, otherwise known as AHL, which is just a type of signaling molecule. So like once again, the little red pentagon looking things in the previous picture, the chemical that goes in and out of the cell. And this signaling molecule is involved in quorum sensing and is common among proteobacteria, which is just a group of bacteria. You might know E. coli, which is a part of that group. AHL promotes the production of this protein called LAS-R. And LAS-R activates a bunch of genes that code/produce a lot of extracellular factors. And these extracellular factors are just proteins and enzymes that will leave the cell. That's where the extracellular part of this comes in. So AHL will promote the production of protein LAS-R, and LAS-R will promote the production of extracellular enzymes and proteins. And these extracellular enzymes are known as public goods because they leave the cell and they're shared by a community. So they're kind of like public to all the bacteria in one environment. And we'll be referring to these enzymes as proteases. So a little bit more about these like mutants/social cheaters. So the growth of P. aeruginosa with casein, and casein is just a type of growth medium, so just a carbon source, energy source for this bacteria. The growth of this Pseudomonas requires LAS-R proteases, which encourages the emergence of social cheaters. So the growth of this bacteria produces a lot of LAS-R enzymes, and all of this extracellular enzyme that is shared for the community that encourages the emergence of social cheaters. This emergence of social cheating or emergence of mutants in a community has the potential to cause this thing called a tragedy of the commons, where the population gets so overtaken by mutants and cheaters that it's ultimately unable to provide itself. So if we think back a little bit to what social cheaters are, they don't produce these proteases to share with their community, they only uptake it. And so once there are too many of these cheaters, there aren't enough bacteria there to produce any of the enzymes and proteins needed to survive. And so ultimately, they are not self-sufficient, and the colony will eventually end up dying. So this kind of leads us to this question, well, so social cheating is really dependent on public goods, those LAS-R proteases and enzymes. So can quorum sensing control, if we can control private goods through quorum sensing, can that control social cheating in a group of cooperators? So we can kind of have a foundation for experiments based on previous literature. And we have a lot of literature already on quorum sensing control of adenosine metabolism. So adenosine is a private good. you need enzymes specifically to break down adenosine that are different from casein. And these enzymes are not shared within the community. The bacteria has to produce them itself and to be able to break down adenosine and metabolize it. So this kind of forces the bacteria to privately make its own goods and to be able to survive. So the experiments in this publication were focused on whether protease secretion, Lazar protease secretion, which is the public good, and adenosine metabolism, which is the private good, can inhibit social cheater invasion. So if we focus quorum sensing on a public good, on shared enzymes to the community, will that inhibit social cheating, which we kind of see doesn't? But will adenosine metabolism, which is a private good, if the bacteria has to make its own enzymes, it cannot cheat off of other bacteria, will that inhibit social cheater invasion and ultimately inhibit a tragedy of the commons? So before I go on, I know that was kind of like a lot of definition and a lot of context to unravel. So I just want to make sure that everybody has like an okay understanding of what's going on before I go on to the actual like data and the testing. Okay, I think everyone is good. So I'm just going to keep going. You can stop me if you have questions. So there were three main arguments / points that the researchers made in this publication. The first being that adenosine restrains social cheater growth, that this idea that quorum sensing a private good restrains social cheating. The second being that cooperators will purge social cheaters in an environment where there are cooperators and social cheaters, that they'll push those social cheaters out, and that more restrictive resources leads to more social cheating. And I'll elaborate on these a little bit more. So the first that we're going to look at is that adenosine restrains cheater growth. So I know that the graphs, the letters are out of order, but they were kind of ordered kind of weirdly in the publication. I just wanted to organize it so that it's a little bit neater and easier to understand. So the first thing I want to go over is the y-axis and making sure that we understand what exactly we're measuring here. So it says percent protease negative, which is really just a fancy way of saying the percentage of cheaters. The protease negative portion being that there aren't there are less proteases in the environment, meaning that there are less cooperators making those proteases and more cheaters taking up those proteases. So really the percent protease negative really means percentage of social cheaters, to put it simply. And also in each of these cases, there is this black line here in the first graph and the black line in the third graph are casein only. And in the rest of the cases with adenosine, there is a consistent and constant amount of casein with the adenosine. So it's adenosine and casein together with just varying levels of adenosine. So just wanted to set up those graphs for you a little bit. So if we look at the graphs, we see that with all of the high percentages of adenosine, we see a really low percentage of social cheaters. But what's really interesting to note is that only when we get past 0.5% of adenosine do we really see any impactful change. So if you look at graph A, we have 0.75% adenosine and 0.9% adenosine, and they're virtually at zero with just a little bit of cheaters here and there. Same with on graph E, we see that at 0.9% adenosine, it's pretty much at zero. But when we go at 0.5% adenosine or below at 0.25% adenosine, we see that there's actually a pretty substantial amount of social cheating going on. In case A, it's pretty much the same, pretty similar to when it's just casein. And in the last graph, we see that there's a pretty substantial amount. Same with D. And also noting that there in graph D, 0.75% or 1.5% of adenosine had no social cheating at all. So these graphs are all very similar. They're just exploring that threshold of where does adenosine really inhibit cheater growth. So here we see that it's anything above 0.5% and anything stronger than that will always inhibit cheater growth. But anything below will still allow for a pretty substantial amount of social cheaters. So there are 3 more experiments to look at, and they're more like controls to really rule out any factors. So if we look at this graph here, graph B, this is a control graph. If we look at the y-axis, it says OD600 part of subculture, and that's just a fancy way of saying density or population concentration. They used a very particular machine to measure, and OD600 just happens to be that measurement. But really what you need to focus on is that over time, this graph kind of plateaus together, meaning that over time, whether it has just casein or different levels of adenosine, that we see the same density and population growth over generations. So this kind of rules out the idea that, oh, maybe we have less cheaters in our population because our medium just doesn't grow that well in adenosine. So it rules out that idea that this is like a medium related problem with generational growth that isn't specific to cheaters or cooperators. Moving on, this was a test on an adenosine plate. So this plate is adenosine only. We see here these kind of interesting numbers and combinations. Those are just different strains of Pseudomonas. They just wanted to see if this is specific to Pseudomonas aeruginosa, or if this is kind of like a more general thing. So this WT that you see on the left side here, that's just stands for wild type. It's an unaffected, unmutated, just normal, natural bacteria. And QS mutant stands for quorum sensing mutant. So these are mutants that are not able to quorum sense. So they're not able to share these receptor or these chemical signals and receive those signals and act as a normal quorum sensing bacteria would. So in all cases, we see that the wild types did grow. So there's nothing wrong with adenosine as a growth medium. We see that the normal Pseudomonas is able to grow in this medium, and it really doesn't have any troubles with it. But we do see that generally there is no growth when there is when their ability to quorum sense is taken away, with the exception of BE177. So this kind of tells us that quorum sensing is really important for the growth of this bacteria and for their ability to survive. So this tells us that quorum sensing is a really integral part to bacterial growth. And this rules out that quorum sensing may not be significant. It definitely is, with the exception of BE177, like I said before. And if we look at graph B, that's kind of supporting this idea that it's able to grow fine on this medium, and that it isn't specific to one or the other. And that was one of the bigger concerns, was that, oh, this might be due to a growth medium, but it's really not. Any questions on any of these tests? Do you want me to go over anything? I'll pause. Okay, I'm going to keep going on. And there'll be questions on the end if you have any lingering thoughts about them or questions. So the last test is really interesting to me because it explores this idea that maybe something quorum independent produces the same result. So we know that adenosine is quorum sensing related. It is controlled, or adenosine metabolism is controlled by quorum sensing, and therefore it serves to suppress cheating. But what if we explore that something that isn't quorum sensing dependent? So we know that casein metabolism is quorum sensing dependent, and it encourages social cheating, and that adenosine metabolism is also quorum sensing dependent, but it serves to suppress cheating. So we've explored things that are quorum sensing dependent, but let's see if something that is independent of quorum sensing produces any result, actually. It doesn't even have to be the same. Researchers in this publication explored that by replacing adenosine with D-glucose, which is metabolized independent of quorum sensing. It doesn't happen whether there's a large population or not. It just happens no matter what. It always will be metabolizing D-glucose. So even when the glucose constituted at least 75% of the available carbon, as we see here, cheaters still emerged and reached a fairly substantial portion. So we see here at the end, they're at roughly the same amount. And this kind of tells us that even if there is something quorum independent, we still see cheaters emerge. So we know that this has to be related to something quorum dependent for the suppression of cheating. And that's why we looked at adenosine, because adenosine is linked to quorum sensing, and it is dependent on that control. And therefore, we see more cheater control with adenosine. So this is just really overall telling us that the way to control social cheating is through something that is quorum dependent. So that next point that I wanted to go over was the idea that we purge cheaters. And this one's a little bit shorter. There's only 3 experiments done. And there were 3 different cases that they explored to sort of figure this out. So they started with an adenosine-rich medium, an LB broth, which is a super nutritious medium, which has a lot of energy sources, a lot of carbon sources. Essentially, the bacteria shouldn't have a problem with getting its carbon. And also the last case being that there was an addition of glucose to a casein and adenosine medium. How they measured this was through a competitive index that they created. Basically, this competitive index is describing how hard it is for cooperators to be getting their resources, so how difficult it was for them, how much they have to compete with the cheaters. So let's start with the first case, A, in an adenosine-rich medium. We see that there's a pretty good decrease in competition for cooperators. I forgot to mention that all these experiments start with both wild types and cheaters in their system, so both cooperators and cheaters. But over time, we see that there's less competition for cooperators, and we can kind of make this assumption that, the cheaters that were already existing in this in the initial colony are somehow getting purged out because we're seeing this decrease in competition. If we look at C, that kind of enforces that idea, but I'll get into that a little bit later. It's a little bit different of conditions. If you look at graph B, in LB broth, we see no change in competition for the cooperators. And this is because there is just such an abundance in carbon sources and energy sources that the competition between the cheaters and the cooperators isn't really affected very much. Both the cheaters and the cooperators are able to get the nutrition that they need, and they feel no competition with each other because the environment already provides all of the energy source that they really need. So that's why we don't see any change in competition. This tells us that in an environment where there's enough carbon source, quorum sensing really isn't super beneficial as it might be when there are limited resources, when cooperation is a lot more important. So in graph C, this is with the addition of glucose to a casein and adenosine medium. Once again, they just wanted to see whether a quorum sensing independent medium might change how competitive it might be for the cooperators. But we see that there really is no effect. If something is quorum independent, we don't really see any change in how the cheaters are going to be like super affected. So in the presence of adenosine, glucose won't change the way that adenosine is going to control social cheating. Same with how the addition of glucose to casein isn't going to change the growth of social cheating. So we still see that nice decrease over time in competition for cooperators, even if we have the addition of glucose. And that's because glucose is quorum independent. So B and C are really showing how quorum sensing is kind of important in certain circumstances to the bacteria. B being that quorum sensing is important when you need shared energy sources, and C being that social cheating and controlling it is really reliant on quorum sensing. So now that we've discussed the results, we kind of need to explore how that happens. So overall, social cheaters were actually forced to cooperate by restricting the carbon and energy source, which restricting the carbon and energy source was through the exchange of casein for adenosine. So rather than really being like pushed out of a colony, they actually had to change to cooperate. Otherwise, they really wouldn't be able to survive. So because there's a presence of adenosine, these bacteria, these cheaters, have to produce adenosine hydrolase, which is a type of enzyme that breaks down adenosine for the bacteria to metabolize it. So because there's a presence of adenosine and that's their energy source, they have to be able to break it down and be able to use it for nutrition. Adenosine hydrolase is a private good, so it's made by an individual bacteria. You cannot get adenosine hydrolase from a community of bacteria. It's individually made, and it's controlled by quorum sensing. So the bacteria will get all these signals to make adenosine hydrolase, but they don't send it out of the cell to share with others. They just get this signaling that, okay, it's time to make this enzyme so that I can survive. So because of this presence of adenosine, they're literally forced into cooperation. Otherwise, they will end up dying. So through experimentation, it was judged that adenosine and adenosine proteases, which again is an enzyme, that production, strongly selects against social cheating. And on to our last point, more restrictive resources leads to more social cheaters. So to kind of explore this idea, what the researchers did is that they replaced casein as a sole source of not only carbon, but also nitrogen. So in the previous experiments, they kept the nitrogen sources the same and unimpacted, and instead added adenosine as another carbon source. So while exploring this idea of replacing carbon and nitrogen sources with casein, they hypothesized that this would probably increase the demands for proteases, and therefore the cost of cooperation would increase. So without nitrogen, which is the NH4Cl that P. aeruginosa uses, the cheaters emerged and actually constituted about 80% of the population. So it's a fairly large and really significant part. In previous experiments with adenosine and casein, they saw percentages of cheaters probably at most probably like around 60%, typically around 40. So 80 is a huge jump from that. So we can see that this replacement of nitrogen with casein had a huge impact on the amount of social growth, cheater growth that we saw in the population. The cooperators produced actually about 10-fold the amount of public goods of their parents. So from the previous generation of bacteria to the next, there was 10-fold the amount of production in public goods. So we see that this restriction in nitrogen had a really, really big impact on the way that these cooperators were working and how many cheaters we saw in the society, in the community. As a result, the cultures turned visibly blue as a result of piocyanin production, which is like a pigment. Piocyanin is quorum sensing regulated, so it senses when the bacteria is under a lot of stress as a community, and then is released into the community again. And it's a metabolite that fights all Pseudomonas competition. So this tells us that the production of piocyanin is telling us that there's a huge amount of stress going on in this community of bacteria, and that they feel a lot of competition with something, and it might not even realize that it's its own species of Pseudomonas, but that there's just too much competition going on that's taking up the goods that it's producing. So this increased cost of cooperation, which is put on by these nutritional conditions, leads to a greater relative benefit for cheaters. And this can lead to a tragedy of the commons, because at some point there's going to be too many cheaters and not enough cooperators to uphold the system. And consequently, the colony will end up dying. So just to start with these tests, on the y-axis of graph A, we see that they're measuring the amounts of piocyanin in a community and how much is being produced. And once again, we see percent protease negative, which is the percentage of cheaters in a community. If you look at all these different shapes on all these graphs, those are different colonies, different trials of the species P. aeruginosa. They just did it multiple times. And this dotted line here is one trial with nitrogen, just as a comparison. And same here with this red, that is with nitrogen. So they wanted to compare and visually show you how much really the exclusion or inclusion of nitrogen really does on a community. And we can also see this comparison between the two colors of the colonies when there is a lot of piocyanin and when there isn't. So if we look at graph A, we see that when there isn't nitrogen as a nitrogen source, the piocyanin levels are really greatly increasing. There's just a lot of stress on the environment, and piocyanin production is really high. Similar to C, we see that when we look at these black lines, we see a lot of cheater growth, even close to getting to 100%. So like I said, that 80% that we talked about before, we see this huge growth in social cheaters. And in comparison to when we do have nitrogen, we see this kind of like smaller line. There's always going to be some kind of competition with casein present because adenosine isn't there to inhibit social cheating. So there is a little bit of stress, but not quite as much as when nitrogen is taken away. Same here. There's going to always be a little bit of social cheating with casein, but not as much as when casein becomes both the carbon and nitrogen source. So we see this really huge impact on how impactful nitrogen is, and that if we limit as many resources as we can, so we're limiting our nitrogen resource, that it's going to have an even larger impact on social cheating because it is more beneficial for a community quorum sensing action like producing casein enzymes for cheaters than it is for cooperators. So to move away from the lab experiments, because they are in a really controlled environment and are super specific and need to be that way because we want to know exactly what is causing what and not based off of correlation. We can explore what really happens in nature, right? Because there has to be a natural way where a community is able to purge social cheaters or regulate it in some way. So the researchers of this paper kind of hypothesized that there might be other mechanisms by which evolution of social cheaters might just be restricted. So there just might be other things that kind of evolve in a way that social cheaters are kind of selected out. This is related to that next point where just the dispersal of individuals to different habitats where cooperative behavior is really important, just kind of naturally selects against social cheats. And that's just one of the possible mechanisms that they proposed in that first point that I put down. And this is actually seen in social amoeba Dicotyledon discodium. So you might see in somewhere in nature, you might see like a really slimy kind of like bacterial culture, and that slime is an example of quorum sensing response. And that slime is really important to protecting bacteria, and it only can happen if there's a lot of cooperation between them. So that is one of those examples where cooperative behavior is just too important to allow for the existence of social cheats. And there's a lot of examples, but this is just kind of like one of those smaller examples. And some takeaways and how we can apply this to our lives in the modern day. Being able to understand quorum sensing and controlling cooperation can actually allow us to manage bacterial populations in natural settings. So there's bacteria all around us, and in order for us to manage those populations of bacteria, we kind of have to understand how they work and how they work not only as an individual, but also as a community. And quorum sensing is really big it's really common amongst a lot of bacteria. And so being able to understand that process will help us manage those populations and, um, you know, kind of help benefit us without really doing too much to bacteria. Um, Another thing is that social cheating can actually hamper the use of synthetic quorum sensing circuits for like, biotech. Um, we want all of our biotech to be as efficient as possible, and social cheating might inhibit that efficiency and how well it might work. So being able to understand quorum sensing in that sense could help us keep biotech efficient and prevent it from kind of like slowing down or inhibiting its actual application. And this is where I am. We'll stop for questions. I am done with my presentation. If you guys wanted me to go back to any slides or go over anything again, this is your time. Okay.
Hi, Deborah. I had a question.
Okay, great.
If you go to your first figure where they're
Sure.
plating bacteria on the plates,
Yeah, here.
yeah, that one. I was wondering about that BE177 line.
Oh, yeah.
Why is that particular mutant growing when all the others aren't on adenosine media?
Yeah, so they didn't really explain exactly why, but what I kind of hypothesized from that is that quorum sensing just might not be as integral to this particular strain as the other strains. So it might just be able to live in a colony that just is able to metabolize the adenosine on its own without having to work with other bacteria in the community. So some strains are just kind of different like that. Like one colony just kind of mutates and it, seems to be successful in certain environments, and they just happen to be that way. So bacteria are kind of funny like that. But generally speaking, quorum sensing is super beneficial. So most strains don't do that, but there will always be like that random instance where they just grow and you're kind of like, what? But yeah, it just happens through mutations and evolution. So I hope that answers your question. Yeah. Does anyone else have any questions? Okay. I'm going to take that as no questions. So if you guys have more questions later on, the slides will be back on the Facebook page. You can also email Professor Dandekar. He was the PI and the one who led this research. And all of this information is available. Oh, I think there's something in the chat. Oh, thank you. So yeah, you can reach out to him if you have any questions about this paper or about physician stuff. So he's available online. Does anyone want to volunteer for our next journal club? And Daniel and I were kind of discussing that instead of doing like a full presentation, if you want to do like a short little spiel or like elevator pitch about your research that you're involved in or interested in, that also works to kind of encourage some people. I don't know if
Yeah,
anyone's interested.
for the next journal club, we're gonna try to make it less demanding for individuals to make an entire presentation, and instead we're gonna shift over to a more like of a 5 minute elevator pitch talk. So that's where you give like a summary of your research in a concise manner. This is very useful because if you're applying to grad school or applying to MD, PhD, you are going to get the question, describe your research to me. And in order to do that, you're going to have to prepare like a 2 to 5 minute talk um, and just discuss your research in a very concise way. And I was thinking maybe people could make one PowerPoint slide and give a 2 to 5 minute talk. It will
Yeah.
be more inclusive so that more people could give their research talks. And it will also be less demanding on individuals to make an entire presentation.
Yeah.
So,
Or if you've done a research symposium before and you have like one of those poster flyer thingies, I kind of forgot what you call them. You can share that with us too.
I'm down to do it.
Who was that?
Danny.
Okay, Danny. Yeah.
Oh, Ridley, because I think there's 3 of us now.
Yeah, there's a lot of Danny.
Yeah,
yeah, a little bit too many Daniels in this class.
exactly. Critical
Okay,
mass.
cool. Yeah, and it can be just like that elevator pitch that we were talking about.
Sounds good.
Thanks for volunteering.
And when are you guys thinking that you want that to be?
Probably in like a month.
Sounds good to me. Yeah.
Yeah. Okay.
Probably sometime next quarter.
Yep.
Yeah.
All right. And if you're new to this club or the meeting and you want to learn a little bit more about us, you can check out our social media. We have a Facebook page and an Instagram that we're most active on. So yeah, we would love it if you would join us. That'd be great.